Hello and welcome to the second lecture of human biology. We will be talking about cells and answering the question, what is a cell? So all organisms are made of cells, hundreds of millions and billions of cells. I do want you to know that cells are the fundamental unit of life. We did discuss that in the last lecture. They're going to be incredibly small, and you're usually going to need a microscope in order to see a cell. There are a few examples of cells that you don't need a microscope to see, and we'll go over that. I do want you to understand this term here. It is cell theory. It is that nothing smaller than a single cell is considered to be alive. The structure of a cell is directly related to its function, and new cells arise only from pre-existing cells. So I do want you to understand all of these concepts here. If you want to think of them like this as being like part 1, part 2, and then part 3, and that all has to do with something called cell theory. Now, just to kind of throw back to the last lecture that we had, I do want to remind you kind of where we are. This class is essentially going to go in the order of organization of an organism, starting with cells, which is what we are going to discuss right now. So we do want to talk about the anatomy of specifically an animal cell. Plant cells do look similar, but a little bit different than an animal cell, but today we are focusing on an animal cell. So we are going to go over all of these various structures here, which I do want you to know that they are called organelles. I want you to think of them as the organs of a cell. It's kind of a weird way to think of it, but a cell is kind of like a person or like a human. They have all these various structures inside of them that make them be able to perform the functions that they are destined to do. So we will talk about the basic anatomy of an animal cell. So the first terms I want you to know are cytosol versus cytoplasm. Cytosol is going to be just this blue fluid in here, and it's not always blue, but for the sake of this structure here, we are going to call it that lighter blue fluid throughout. And this is going to contain water, salts, and enzymes. So enzymes are going to be things that digest other things. So kind of like stomach acid is kind of a good idea of an enzyme. Cytoplasm is going to be cytosol as well as the organelles themselves. So it's pretty much everything within the cell. And then that's excluding this dark blue ring on the outside. I'm not doing my best at keeping it, my drawing, not including that dark blue. But the dark blue is going to be something different. But just know that cytoplasm is going to be the fluid within the cell, the cytosol, as well as all of the organelles. So first and foremost, we are going to start with the nucleus. That is this big pink egg here in the middle within that black box. The nucleus will surround the nucleolus, which is the yellow portion here, and stores genetic information. It is surrounded by the nuclear envelope. That's what this light pink is. That is the nuclear envelope. And it will contain chromatin. Chromatin is a combination of DNA molecules and proteins that make up chromosomes. Chromosomes should be a term you're potentially familiar with. You have 23 pairs of chromosomes. So in total, you have 46 chromosomes. And any deviation from those chromosomes is going to change your genetic makeup. So, for example, chromosomes are going to affect what you look like and how your body functions. We will talk about chromosomes more a little bit later in the semester. But for now, just know that chromatin is just that combination of DNA molecules and proteins. And then next we have our nucleolus. That's this little yellow guy here in the middle. And it is going to contain ribosomal RNA that is going to be produced and combines with proteins that form ribosomes. And ribosomes are these little circles kind of out and about here, as well as attached to these structures here. And we'll talk about them as well. So next up is an organelle that you should be quite familiar with, which is mitochondria or a mitochondrion. And very simply, it is the powerhouse of the cell. It is going to produce something called ATP, which is adenosine triphosphate. But what I want you to know more specifically is that it is energy. I do want you to know that inside the mitochondria is a small circular chromosome, so that is what this is showing here, this little kind of wiggly line. I always think mitochondria kind of look like sausages with a little bit of mustard or ketchup on them, kind of swiggled on. But mitochondrial DNA is passed from mother to ovum, which is also known as the egg. And this can be used to examine maternal family ties. So as far as DNA goes, you will always receive your mitochondria from your mother. So do know that. So your mitochondria is going to come from your mother, never from your father. Next organelle we're going to talk about is the lysosome. This is going to contain a digestive enzyme. Like I said, those enzymes are going to be kind of like stomach acid. So what I want you to think of the lysosome as is like the stomach of the cell. It is going to degrade proteins, carbohydrates, and fats into the cytoplasm. This is called autodigestion. So again, the lysosome is much like the stomach of the cell. It's going to break things down so that the cell can use them. Next up is the endoplasmic reticulum and ribosomes. So there are two types of endoplasmic reticulum, which from here, just because it's a tongue twister, I'm going to call it ER. So you have rough ER and smooth ER. The rough ER is going to contain ribosomes. So that is what we have here in this box. That is going to have those little white dots attached to it, which are the ribosomes themselves. And you can see in this box here, this is just a ribosome by itself. Proteins are going to be synthesized and enter the interior where processing and modification begin. Some are incorporated into the membrane while some are exported, and this is talking about the ribosomes specifically. Ribosomes themselves are made of proteins, and they can be attached to the ER or are free floating in the cytoplasm like we see in this graphic. Now, the one thing that I don't like about this graphic is that it doesn't really show smooth ER very well. In fact, I don't believe it shows it at all. But just know that the smooth ER looks incredibly similar to the rough ER, just no ribosomes. The purpose of the smooth ER is to remove harmful substances and synthesize lipids. They're going to wrap them in membrane envelopes. And it's going to form transport vesicles to move large molecules around the cell. Next up is our Golgi apparatus or Golgi complex. Sometimes different books call it different things. And that is the structure, and I'm almost blocking it, but hopefully in your notes you can see it a little bit better. I kind of think it looks kind of like a Wi-Fi symbol or I don't know, something to that effect. Just the way it looks is going to be kind of like this. It is going to receive packages from the smooth ER, so this is going to be packages of proteins and lipids, and it is going to process or modify these proteins and lipids. For example, it could add a sugar chain to either, which is going to create glycoproteins or glycolipids, so sugar proteins or sugar lipids, which, just so we know, lipids mean fat, and then send that off to the plasma membrane. Now, we didn't really talk about it very much, but I kind of hinted at it. The plasma membrane is going to be this dark blue. Plasma membrane. Or send to proper destinations around the cell, those little packages. We also have secretory vesicles, and I don't believe I have anything on this graphic that shows that, but I do want you to know what it is. It's going to contain materials produced in the cell. It's going to be formed by the Golgi apparatus and is secreted by a process called exocytosis. And I do have a slide that talks about exocytosis a little bit later. Centrioles, probably one of my favorite organelles, is going to facilitate the movement of chromosomes during cell division. And we'll talk about cell division here in a little bit. To me, they look like churros, and with this graphic, the way that they are colored, they look kind of like licorice. Microtubules are going to be those little sticks. This is going to support the cytoplasm and assist in cell division and form components of cilia and flagella. So that's going to, as far as cilia and flagellum are concerned, this is going to help with movement of the cell. And when it comes to cell division, that's going to be the cell dividing, oddly enough, and forming another cell. You can also kind of think of them as the bones of a cell, since they are for support. But don't think that they're just these very rigid structures, but they kind of act a little bit like the support that bones give your body. So on to cilia and flagella. Up here in the top picture, where it has the number 3, this is cilia. So these little hair-like structures. They can line your respiratory tract, which serve the purpose to sweep debris within mucus up and out. So this is actually a slide of a few cells. You can see the nuclei within here, as well as goblet cells. So this is a goblet cell. So when I said before, that's not a nuclei, that's a goblet cell. And what these goblet cells do is produce mucus. So this is going to be cells that you would find in your trachea. So the tube that leads from your mouth to your lungs. And what happens is it produces this mucus that pops out to the outside of the cell. So this would be sort of like your throat. So that tube. So the empty space. This cilia is going to move in kind of a wave-like pattern and trap debris inside of the mucus, and then you'll cough it out or you end up swallowing it. It just kind of depends. But these cells are kind of interesting in their shape, and we'll talk about cell shape here in a bit. Microvilli are going to be extensions of the cell surface, with many on each cell. So it's kind of like this, but then you have smaller structures coming off. So this structure here in the middle would be a villi with microvilli coming off of it. You'll find this pretty exclusively in other places as well, but the biggest place you can find them is going to be your digestive tract. But it's going to increase the surface area of a certain cell. So instead of just having, just the outside here, you have it going up and around, up and around, up and around. So you can increase the surface area for more absorption of materials like water or nutrients. Then last and certainly not least, you have a flagellum. A flagellum is going to be this whip-like tail on a cell. And the example I want you to think of is a sperm cell. So this right here is a sperm cell. And it will help in the movement of that cell. Both of these actually do. I don't know that I stated that. Now moving on to the plasma membrane. So remember, this is the outside of the cell, that dark blue. It is the outer layer that regulates what enters and exits the cell. It is going to be composed of a phospholipid bilayer, so it's a sandwich of phospholipids which are selectively permeable. Now, what this term means, selectively permeable, is that it is picky about what it lets in and what it lets out. So you have these various channels down here that are going to let potentially proteins, specifically for this example, this little channel is going to let proteins in and out of the cell. Other things are going to need to be assisted in and out of the cell. There isn't always just a channel that allows things to go in and out. So speaking of things going in and out, we're going to talk about molecule movement. There are various types of movement that a cell can undergo, and this is just moving things in and out of the cell. I do want you to know all of these terms, what they mean when it comes to your test or your exam. I will use the exact verbiage from my slides. So do know that diffusion, the big one, is going to be just movement of molecules from an area of high concentration to an area of low concentration. So what that means is that you have more of something on this side and less of something on this side, and they're going to move until they are balanced. So an area of high concentration is going to move to an area of low concentration. Hope that makes sense. Passive transport is going to require no energy, so make sure you know that passive means no energy, very important, to move molecules across the plasma membrane. Then you have facilitated diffusion. So these are membrane proteins that help facilitate movement across the membrane. And then active transport. It's going to use proteins to move molecules against the gradient. So areas of low concentration to areas of high concentration. I want you to think of active transport as being kind of the opposite of passive transport as well as diffusion. So it's areas of low to high and from areas of high to low. Next up, we have water movement specifically. So before it was just regular molecules, now we are moving on to water movement. And this term is going to be osmosis. Osmosis is passive transport of water across a membrane. We have a couple terms that I want you to know that are involved with water movement, and that is tonicity, and that is the ability to make water move in and out of the cell. So tonicity is basically like just a descriptive word. You're asking about the tonicity of it. And then when it comes to tonicity, we have three terms I want you to know. So isotonic is the same amount of water inside and out of the cell. So that's this middle example here. So the same amount of water is going in the cell as well as leaving the cell. Hypertonic is going to be where the cell shrinks as water leaves. So that's this first guy here. So you can see that the cell is shrinking in. And then the last is hypotonic. This guy over here on the right. Water is coming into the cell and making it expand. So a good way to remember these various types of tonicity is that isotonic is kind of unique in that it has an I in it. It's not quite as confusing, but hypertonic is going to have an E versus hypotonic has an O. Think of O as being like an O, it's swelling, turning into an O kind of thing, and hyper, an E, kind of looks like a shriveled up letter, if that helps maybe. Then here we have exocytosis versus endocytosis. As the names imply, exocytosis is going to mean exiting the cell, and endocytosis is going to be entering the cell. And anytime you see this word, cytosis or cyto, it is referring to the cell. And then you can see this nice example of endocytosis of food particles coming into the cell. We have a food vacuole as well as vesicles that are transporting this cell through the cell, or I'm sorry, transporting the food through the cell, and we have our lysosomes coming in, releasing the digestive enzymes, and then we have a digestive vacuole, which is going to remove the particles that you don't need for the cell through the process of exocytosis and eliminating those products from the cell. All right, moving on to the cell cycle. So this is the last little section here on cells, and then the next lecture will be over tissues. So let's ask the question, how does damaged tissue replace and repair itself? So if you got a cut on your arm or on the inside of your mouth or wherever, those cells are damaged and they need to be replaced, but that also needs to be repaired as well. So let's figure out how cells are able to do that. So some terminology I want you to know as far as we go. I want you to know the term diploid or two-in. This is an organism having a complete set of chromosomes, 23 pairs. This is also called a homolog. Haploid, or one-in, a single in, is possessing half the normal number of chromosomes. And then I want you to know that gametes are haploid. Now, this is a term we maybe don't know, but gametes are sex cells, such as an ovum. Sorry, that kind of looks like bad handwriting. Or a sperm. Those are gametes, and they are haploid. They only contain half the normal number of chromosomes. So they only have 23 chromosomes. A diploid creature would have 46. Okay, so 23 pairs equals 46. And then tetrad is going to be paired or duplicated chromosomes visible at the start of meiosis 1. And that is the process that we will be talking about here in just a moment. So cell division, it is the formation of daughter cells from a single parent cell. I do want you to know the difference between these two types of cell division, mitosis and meiosis. Mitosis is going to produce two genetically identical daughter cells. This is going to occur in tissue repair. So if you get a cut and your tissues need to basically repair themselves as well as produce more cells to cover that cut, as well as embryonic growth. So from the time that you are a little zygote, where a sperm and an ovum come together to create a fertilized egg, as those cells grow, they are using mitosis in order to create more cells, which will eventually create a fetus or an embryo. And then we have meiosis, which is cell division that only occurs in the gonads, so in an ovary or a testicle. And these are going to produce haploid gametes. So remember, that's only 23 chromosomes, and they are sex cells. There are two successive stages of meiosis. It is meiosis 1 and meiosis 2, and we will discuss those a little bit further. So here, just to make sure we're all on the same page, we are just talking about how a cell divides and produces more cells. I do want you to know these various stages of meiosis. We have interphase, which is the non-dividing phase. That's very important. This is where the cell is just going about doing its normal functions. It's just being a normal cell. Nothing's really happening, except potentially the chromosomes are replicating. So remember, the chromosomes are going to be within the nucleus. From interphase, we're going to enter prophase 1 because we are in meiosis 1. This is going to be where it is homologous chromosomes form tetrads and exchange segments. And you can see in this little graphic down here at the bottom, as well as up there at the top, that we are having this little crossover happening and we are exchanging that information. So this is going to increase genetic diversity and ensures all 4 daughter cells are unique. This is important because you don't want everybody to be exactly the same. Otherwise, if something goes wrong and there's a malfunction within that genetic coding, then that problem gets passed on and passed on. So by splitting it up and exchanging that information, we have a higher chance of something good happening rather than something bad. But this is only done in meiosis. Remember, we are talking about sex cells here. Metaphase 1, we are going to have the tetrads align in the center of the cell. Anaphase 1, we're going to have homologous chromosomes separate and migrate to opposite poles. And then telophase 1, the cells will divide and each daughter cell is now haploid, so half the number of chromosomes. These chromosomes are going to be different from the parent cell, and that's a very important thing to distinguish. They are going to be different from the parent cell. They are not identical to the parent cell. They are unique. So here we see metaphase, where everybody's lining up at this plate. It's called the metaphasal plate. Anaphase, we have our homologous chromosomes separating, so they're going their own ways. And then telophase, we're going to have this cleavage site, so where the cells are going to completely split and become their own unique cells. Meiosis 2 is essentially where each one of these is going to basically do that again. So prophase 2, each of the 2 daughter cells begins another division. Metaphase 2, we're going to have chromosomes aligning. Anaphase 2, the sister chromatids move to opposite poles, and telophase 2, the final stage where cells divide, cells again divide. So what I want you to get from this is that the final product will be 4 haploid cells. Remember, that means that there's only 23. And they are all genetically different. So this is the take-home message I want you to get from that. Now, I quite like this little chart that I've made for you. I do want you to know it. So be able to recreate this chart. I guarantee it's going to be either on your quiz or definitely on your exam. But be able to know the difference between mitosis and meiosis. And then here is a nice little chart that is also showing the difference between mitosis and meiosis, but you do not need to read or remember. This is for your own viewing pleasure if you so choose to read through it. If it helps you, fantastic. If it doesn't, ignore it. So that should conclude our lecture here on cells and cell division. If you have any questions, don't hesitate to message me. Other than that, our next lecture will start on tissues, which is just groups of cells.